Good morning, good afternoon, and good evening, everyone. Welcome to FABI International Seminar Series. And if you don't know me, my name is Neriman Yilmaz, and I'm a postdoctoral fellow here at FABI. And together with Dr. Marcus Wilken, we chair the FABI International Seminar Series each month. And this month, our speaker is joining us from Italy, Dr. Claudia Coleine. And Claudia received all her degrees from the University of Tusia. And during her postdoctoral fellow research, she was the principal investigator of a project funded by Italian National Program for Antarctic Research. She's currently currently received a very prestigious Marie Curie Global Fellow grant. And she will untangle the microbiome of US drylands and defining those physiochemical conditions that still allow active life, even at the extremely low water regimes. So Claudia and I personally met during the Unite workshop in 2017 in Aberdeen. And since then, I'm a big fan of Claudia and follow all her work throughout all her publication. And today, as in her topic, she will take us to the very extreme environment. So Claudia, thank you again for joining us and giving your seminar, and we are looking forward to your presentation.
Thank you, Neriman, for inviting me again, and good afternoon to everyone. Thank you for attending this seminar. As you said, I did my PhD in the University of Tuscia, working with Professor Laura Selman, who is involved in Antarctic research since many years. And in a few weeks, I will move to the USA to start a Marie Curie Global Fellow project. But I will mention it later on this presentation. Just a few words to let you know that the microorganisms from extreme environments from this topic fascinated me since my master's degree studies when I work in an astrobiological research context. But today we will talk a little bit about microorganisms from extreme environments. But just to start, that as all of us know, microorganisms are almost everywhere on Earth, but even beyond the Earth. Some microorganisms are also found in the International Space Station. And on Earth, we can find the microorganisms pretty everywhere, inside our body, in food, associated with plants, in the ocean. But some microorganisms have also evolved some adaptation strategies to survive extreme conditions. They are found here all natural extreme environments, such as hot springs, dry regions, glaciers, salters, but they can also found in contaminated sites by anthropogenic activities. But I would like to just to point the attention to what means extreme environments. Of course, is something that is related to our anthropocentric point of view, because the so-called extreme environments is an habitat, an environment when one or more physicochemical parameters such as pH, temperature, salinity, or pressure reach values behind those that are considered normal or compatible for with our life, with human life. So, as I mentioned, the microorganisms can be found everywhere, and in particular can also occur in conditions that are concurrently polyextreme. For instance, the hypersaline lakes are simultaneously also alkaline, and in Antarctica, for instance, here in the McMurdo Dry Valleys, of course, there is the aridity as the main challenge for life, but it is also characterized by the high UV and solar radiation, cold temperature, and other several stressors. What is an extreme organism? First of all, extreme microorganisms can be mainly characterized in two groups. We know the extreme tolerant microorganisms that are organisms that can tolerate very well extreme conditions characterized by one of more extreme physicochemical parameters, but they actually prefer and grow optimally under neutral conditions. Conversely, extremophilic microorganisms are those organisms that obligatory need extreme conditions to proliferate, to grow. For instance, we see that, for example, salinity, we have halotolerant that can tolerate salt conditions, while there are some organisms that are accounted as halophile or extreme halophile that require very high concentration of salt. The same thing for the temperature condition. For instance, the hyperthermophile require more than 80 degrees to grow, while psychophile need very, very low temperature. But maybe an important question is to understand why should we study extreme environments? First of all, we need to study extreme environments to untangle the current understanding of life as we know it on Earth, to determine the resilience of life and the potential adaptation to current and changing conditions, for instance, due to climate change and global warming. But we also need to study extreme environments to understand the origin of life on Earth, to define the limit of life on Earth, to highlight the patterns of terrestrial habitability. But most importantly, it's critical to study extreme environments to identify which organisms or which genetic properties an organism should encompass to potentially survive in extraterrestrial environments. And this is something in an astrobiological context, because nowadays we well know that life is not confined in our Earth, but now we didn't find life as we know it in extraterrestrial planets or moon, but study the adaptation strategies and the evolution of extreme microorganisms on Earth, we can speculate about potential life in extraterrestrial environments. At present, it's quite difficult or impossible to travel to around the solar system, but the scientists in the last decades have developed some strategies to answer these fundamental questions. For instance, it's critical to study microorganisms from terrestrial extreme environments that are nowadays called terrestrial analogs. Of course, we are aware that no terrestrial analogs encompass the same condition of another planet or moon, but in some way, many places that are summarized in this map in some way resemble geological, geomorphic, or climatic conditions like another planet or moon. Extremophiles encompasses macroorganisms from the world tree of life. So we have microorganisms from bacteria, archaea, and eukaryota. So far, most of notable extremophiles were considered belonging to archaea and bacteria. It's true, it's quite true. For instance, some members of archaea are the best equipped to survive to hyperthermophile conditions. And here I summarize some notable examples of bacteria and archaea. For instance, the Archeon Metanopyrus candleri can survive up to 122 centigrade and can survive also up to 125 megapascal. Just for a comparison, the maximum pressure that a human can withstand is 6,000 Pascal. But archaea and bacteria are also skilled to survive radiation or radioactive environments. For instance, Deinococcus radiodurans here in the Deinococcus thermosphelum is a test organism for astrobiological experiments and can survive up 15 kilogray and high UV radiation. Or for instance, the Archeon Thermococcus gamma tolerans can survive simultaneously up 95 Celsius degree and 30 kilogray. But among bacteria, without doubt, the cyanobacteria, and in particular, the members belonging to the genus Crococcidiopsis, is among the most tolerant organisms on Earth that is capable to survive up to 113 kilogray radiation. That is very impressive. But as I mentioned, most scientists in the last decades thought that the most extreme tolerant, the stress tolerant organisms on Earth belong to archaea and bacteria. That, as I said, it's quite true, but fortunately, it's now becoming evident in the last years that also eukaryotes are very skilled and equipped to survive extreme environments and extreme conditions, and often they do better than prokaryotes. Among the eukaryotes, the most, maybe one of the most interesting and resistant organisms is this free-handly tiny invertebrate. I just have a video for a few seconds to show that these invertebrates survive unexpectedly a lot of extreme conditions. I can just video. So just a few seconds to show that even multicellular organisms are capable to resist extreme conditions. For instance, the tardigrades resist down to minus 272 Celsius degree to up 150 Celsius degree, resist to vacuum and several kind of radiations. But back to the microorganisms and back to eukaryotes. Among eukaryotes, fungi are, thanks to their morphological plasticity and ecological versatility, fungi are the most skilled lineage into the tree of life. And here I summarized a few examples of stress-tolerant fungi. For instance, fungi are capable, better than prokaryotes and archaea, to resist under a very, very low water availability. Just for a comparison, most of living organisms, including the humans, need to live under a water availability ranging from 0.99 to 0.97. Some bacteria can resist up to 0.7, but this fungus, that's impressive, can resist at a water availability of 0.58. And as I said, the bacteria and the archaea are very skilled to survive in very hot temperature, while fungi are not very skilled to survive in hot temperature, but conversely, they are very, very good to grow and proliferate under low temperature. For instance, many fungi can optimally grow under 0 degree, and a few of them can also grow and proliferate and be active, that's very important, down to -8 degrees. And among fungi, most of them are acidophilic. And again, most of living organisms need to have a pH near neutral, more of them. And while bacteria and archaea are not very good to resist to acidic conditions, fungi can also are capable to survive 0 or 1 pH value. That is very impressive. And they can do by maintaining their cellular cytoplasm near to values near neutral. And they are capable to keep the cellular and metabolic activities like an hibernate state. The last but not the least example I want to highlight today is a melanized fungus, Cryomyces antarcticus, that was firstly isolated from Antarctic rocks in McMurdo dry valleys. This fungus is capable to resist up to 170 kilogram ionizing radiation. Just for sorry, just for a comparison, archaea can resist like sorry. Okay. Archaea can resist up to 30 kilogram, while bacteria up to 15 kilogram. And just to let you know that this fungus, Cryomyces antarcticus, can tolerate 3,000 times the tolerance of human cell and 5,000 times tolerance of Escherichia coli. The tolerance of this black fungus show that this fungus is very capable to resist not only to radioactive environments, but also to salt saturation environments, gamma rays, and thanks to the heat tolerance, it's considered like one of the best eukaryotic test organisms for astrobiological experiments. Cryomyces antarcticus belong to the ecological group of black meristematic fungi. The black meristematic fungi are a polyphyletic group that share several ecological and morphological traits. For instance, meristematic growth, clumpy colonies, polar banding growth, and they share their love for extreme environments. As I said, they are polyphyletic. They mainly belong to Dotidiomycetes and Euroxiomycetes in the Ascomycota phylum. And for black fungi, the melanin, along with other molecules like trialose and carotenoids, allow the spreading of these fungi not only to resist to radiation, but even to resist to dehydration, low and hot temperature, and high or low pressure. These fungi are very resistant, but actually it's not so easy to work with them in the lab, because they have a very, very slow rate. For instance, a fungus can take up months to grow. These fungi are ubiquitous in the world, in the natural extreme environments, such as deserts, and McMurdo dry valleys in Antarctica, acidic sites. Some of them have also been recorded in the polluted environments, and some of them, but fortunately a few of them, are also involved in severe human disease. But despite their interesting ecology, basically we don't still know their environmental preferences and their distribution across the global biomes. And recently, to address this knowledge gap, we did a soil survey from global biomes, including different ecosystems, and we saw that we found that actually soil is a very important reservoir for black fungi. And we found that elevation, nitrogen, and the water availability were the most important predictors for the distribution of these fungi. We also found that the genera Exophylla and Cladiophyllophyra were the most abundant and widespread in global soil. Despite the recent advances, we don't still know the genetic properties and the genetic capabilities of these fungi, and we don't know how they can still live at the age of life. Just to see, now we have around 2,000 genomes available for fungi, and just 60 genomes are available for black fungi. In this context, a couple of years ago, my professor, my PhD supervisor, and the Tush University have been granted with a global project from the Joint Genome Institute in the USA, in collaboration with other 20 countries, to sequence the whole genome of more than 600 black fungal strains to identify the ecology and to sample among the whole tree of life across the main phylogenetic lineages and lifestyles. In addition to the genomes, we also perform some metabolomics and metatranscriptomics to untangle the genetic and metabolic capabilities that govern their success in the extremes. As I said, the most of strains proposed for the project are preserved in the international cultural collection, including the collection of fungi from extreme environments that is now hosted in the University of Tuscia, in my home host institution, where almost 1,600 fungal strains are preserved, and 1,000 of which 1,000 are black fungi. In the years, we also contributed to describe more than 20 new genera and more than 50 new species. And as I said, 1,000 black fungi, but mainly are from Antarctica. So the question could be, where are the black fungi are in Antarctica, and why Antarctica? Just let me introduce a little bit the history of Antarctica. In fact, the Antarctica, the aspect of Antarctica, it's not has the current conditions. In fact, like 600 million years ago, there was the supercontinent Gondwana, and the Antarctica belonged to a subtropical position. And in those days, the bigger vertebrates, the plants and complex living organisms developed. But like 200 million years ago, the fragmentation of Gondwana began, and like 60 million years ago, Antarctica started to move to South Pole. Like 2.5 million years ago, Antarctica reached South Pole, and the environmental condition dramatically changed. For instance, the current conditions we found is cold temperature, of course, the polar cap that covered the Antarctica for a special of 2.5 kilometers, and most notably, the surface is almost ice covered. We have only the 2% of ice free. In particular, in the 2% of ice free that are included in the inner part, in the inner region of Antarctica, called continental Antarctica, the conditions are too harsh. Characterized by aridity, UV radiation, oligotrophy, cold temperature, and conditions are too harsh that are in some way resembling the Martian conditions. In fact, the McMurdo dry valleys that are from continental Antarctica are nowadays counted as the best Martian analog on Earth. In those conditions, these regions were considered lifeless until a few decades ago, when microbial life was found inside rocks. In fact, in those ice-free area regions, rock represent the main substratum for colonization, because when the environmental conditions became too harsh to allow a microbial colonization on the surface, life can find a last refuge inside rocks. This is an example of microbial life inside the rocks that form the so-called cryptoendolytic communities. Cryptoendolytic communities are microbial communities well organized, self-supporting, and complex, where primary producers and consumers are present. For instance, we find black fungi, but even lichenized fungi, algae, and the bacteria, archaea, and even viruses. These communities evolved in genetic and geographic isolation and are among the most adapted and specialized organisms. but in the same time, they are very sensitive to any external perturbation, for instance, due to climate change. So it's nowadays critical to untangle their biodiversity and the potential effect on this vulnerable and fragile ecosystem, microbial ecosystem, that in some way still support the functioning of these dry regions in Antarctica. Continue to study these Antarctic endolytic communities is important for many reasons. I want to just mention a few of them. But firstly, it's important to increase our knowledge on global biodiversity. Because it's constant and continues to describe new fungal taxa, new bacterial taxa from these samples. Again, it's important to untangle the biodiversity and the rock properties governing the success of these communities to define the patterns of terrestrial habitability and search for life during the exploration of rocky planets such as Mars. And the hypothesis is that if Martian life ever evolved on the surface of the Martian current surface, probably life can find refuge inside rocks like it is now doing in Antarctica. My group in Tuscia University is involved in Antarctic research since many years, and since then we work to increase to improve the knowledge on these microbial communities. I want to show you a video of my supervisor, Professor Laura Selman, in a typical day in Antarctica to collect rock samples. I want to just mention that the audio is not very perfect due to some logistic issue, but I hope you enjoy these few seconds of video.
Yeah, I'm Laura Selman and from Italy, and I'm here to look for endolytic life. Here we are on Mount Electra. It's cold, full of snow. So, but as we get off the helicopter, we look for some traces of life, and there is actually. Even here you cannot see anything on the rock, apparently totally sterile, but I see these traces, this weathering, showing what is inside the rock, and this is due to the microbial activity inside the rock that's growing and getting wet and freezing and thawing, and the microbial activity itself detach the surface, the first layer of rock, and show what is inside. So when you see this patchwork, whitish red surface, then you can get sure that there is life inside. So now we crush the rock and look what is in.
Salta via con una scheggia e vola. Ecco, sapevo. Troppo dura per... non è criccata sotto.
Here, just below the rock surface, you see this blackish band. These are already microbes inside the rock. When the conditions are a bit milder than this, because here is very cold and full of snow. You can find even a much better stratified community, with the first this blackish, and then another below that is whitish, and then another one greenish, and they are stratified according with the biological requirements of all the components of the communities. So fungi, lichenized fungi and algae, and then green algae normally. and we could also find some cyanobacteria. Of course, there are also bacteria inside. So it's a very rich community actually. We are sampling colonized sandstone in Finger Mountains. So the rocks, this sandstone is beautifully weathered by microbial activity. So I can get they are colonized even looking at the appearance of the rocks. And then crushing the rock, we see these blackish bands and even greenish bands. This means that a cryptendelic community is inside rock.
Okay, I hope you enjoyed the video. And during the last years, decades, we collect a lot of samples in different locations at different environmental conditions, in a gradient of distance from sea and elevation. We also collected in different sun-exposed rocks. For instance, the north sun-exposed rocks here in the light, where in the left, where in the right, we found the south shady surface. In our works, we found that among the environmental variables considered, sun exposure is the main driving influencing the community composition in terms of species richness, but also taxonomic composition. Not only the sun exposure, we saw that also influence the different metabolic responses. For instance, we found that north sun exposed and south sun exposed communities encompass different metabolic strategies to survive. Here in the presentation, we saw that we found that the melanin pathway that I mentioned before, the melanin is critical for survival and proliferation of microorganisms. We found that in the south exposed communities was more abundant. Recently, we also collect samples along an area environmental gradient, and we use heterogeneous data sets as fungal and bacterial information, but even microclimatic monitoring data and rock physicochemical properties. What we found is that rock properties, in particular quartz cement, porosity of rocks, and abundance of iron were the most important factor associated with the abundance and richness of these communities. And from then, we also sequenced the first shotgun metagenomes from these communities to look more deeply inside the genetic properties and the evolution of these communities. Just a few seconds, because this video can summarize the main findings probably better than me. I hope you enjoy it. And from that study, we sequence more than 100 samples of these communities to establish the first Antarctic bacteria genome catalog. And so far, this catalog contain almost 5,000 genomes, of which more than 2,000 are novel species. Also, we identified and we found that at least one gene coding for the anti-freezing protein is included in the genomic repertoire of all members of the community, supporting the hypothesis that the cold tolerance, these traits, is of paramount importance for survival and proliferation of life in these harshest conditions. But just moving from Antarctica, because the endolytic communities are not exclusive of Antarctica, but are ubiquitous in all drylands, including the hot and the cold, whatever. Ranging from the subhumid region where the water is more available to hyperarid regions where the water is not more available anymore. Drylands are one of the most important biomes on our Earth, based on their extension and their ecological importance. In fact, drylands cover half of the global land surface, support almost the 50% of the globe cultivated systems, and drylands regulate the carbon, nitrogen, and other nutrient cycles. But despite the challenging conditions of drylands, because in addition to water deficiency, of course, there are several other stressors that characterize drylands, such as the high evaporation rates, extremely temperature fluctuation, high UV and solar radiation, and most importantly, soils of drylands are almost always characterized by low nutrients and low soil fertility. But despite the challenging conditions, microorganisms that inhabit drylands have evolved adaptation strategies allow them to survive even in the hyperarid areas. For instance, bacteria are capable to enter in hibernate mode or to produce spore. These both approaches are very resistant, make the bacteria very resistant. Or for instance, other microorganisms have evolved in their cell pigmentation. For instance, in fungi, we found the melanin, or in other microorganisms, we found that they are capable to introduce compatible solutes inside their cell to maintain their metabolic activity under any hibernate mode. Microorganisms in drylands are critical for ecosystem functioning because they form fertile island. They are critical for pedogenesis, providing the backbone for ecological succession of soil, and they are important genetic resource, even of biotechnological potential. In drylands, microorganisms have evolved different ecological strategies. This is according to the water availability. In fact, soil stability and xeric stress are inversely related. And we found in drylands different type of colonization. For instance, from left, we have soil colonization or biocrest colonization in subhumid regions where the water is quite available. Two, the endolytic colonization in the right, where the water is not available anymore. And we found the hyperarid regions. For instance, endolitic communities are important, especially in hyperarid regions, where they represent both the first colonizer of freshly exposed rocks, but even the ultimate colonizer just before the extinction, where conditions are not more permissive. Drylands are notably impacted both by natural changes, but even by anthropogenic activities as overgrazing or intensive agricultural management. So determining and untangling microorganisms inside the rocks and how the ongoing climate change and global warming will affect these microbial communities is important to have to give important insights on the future of these fragile ecosystems that are part of half of the our planet. We know very almost well that soil communities as pretty characterized, influenced by aridity, UV radiation, and we have recently identified some important environmental parameters that are the driving force for the soil communities. But a knowledge gap, an important knowledge gap in the drylands microbial ecology is the endolytic niche. We still don't have a clear and comprehensive picture of the global distribution and adaptation of rock inhabiting microorganisms. And in this context, as I mentioned just in the start, in collaboration with the University of Tuscia, University of California, and the Spanish National Research Center in Spain, we have granted a Marie Curie Global Project to study drylands and in particular rock inhabiting microorganisms of North America. And in a few weeks, I will move to the USA to start the sampling and start producing data. Thank you very much to everyone for attending this seminar. And I would like to acknowledge, to thank all mentors, supervisor, collaborator, and students that worked with me during the years. Thank you. Thank you again.
Hi, Claudia. Thank you so much for very exciting presentation. After showing the Antarctic, I must say that I got the feeling of cold, but then you took us to the dry land, which is, I am sure, like almost 90-degree difference, then it was feeling 45 degrees, and I start to sweat, I must say. So you really make it very lively presentation for us. So I will ask that if you have questions, please raise your hand so I can give you the chance. But before going into the chat box question, I have a question to ask. So like the first question comes to my mind, especially for the Antarctic project. What is their purposes, do you think? What do they do? Because I mean, that place is considered as lifeless. We know fungi has many functions, like one of the biggest reasons is they are decomposers. But sort of if they don't have anything to decompose, so what are their function in Antarctica?
Basically, they live inside the rocks in inactive state, and they have like 1 or 2 days per year when the environmental conditions and the snow, the solar radiation and everything is allowing the metabolic activity of these microorganisms. and in those 1-2 days per year, they are capable to reproduce and keep their metabolism active. Of course, the Antarctic rocks are a very, it's an environment characterized by a very low nutrients, as you rightly said, but there are a lot of mineral, in particular in the sandstone type, because the endolitic communities can be inside the different type of rocks like granite, dolerite, or sandstone. And we found that the sandstone is the best optimal substrate for microbial colonization because of porosity. The porosity of sandstone, on the contrary of the dolerite of granite, that are very much more contact, but the porosity of sandstone allow microorganisms to interact, the microorganisms between each other and the environment, and to take some very low nutrients from substrate or nutrients or minerals.
And last question from mine. With the climate change that is happening, so do you expecting to see any microbial diversity change in Antarctic? And also, are you gonna continuously monitor the region, or because your project sort of finishing, so will you stop there?
Yes, I start from the second part of the question, saying that I show just the data from 4 years of micromonitoring experiments, but the monitoring settings are still in some location in Antarctica, and we will have much more information, of course, over the years. So in the future, we can relate the data from today to over the years. And yes is the answer to the first question, because the global warming and climate change in general, but in particular the global warming, is very dangerous for Antarctica, because the microorganisms I show you are very adapted and specialized. They are specialized to living in those conditions, are not at all competitive. So that means that with the global warming, it could happen that alloctonous species can go in Antarctica and kill the autochthonous, the Antarctic microorganisms that are very resistant but are not competitive. So the introduce of alloctonous species due to the climate change can be cause a severe shift in microbial composition. Yeah.
Thank you, Claudia. And we have a comment from Vitaly Kulka from Republic of Moldova, and he mentioned that because the region where he lives is considered as an extreme condition, therefore he sometimes feels like microorganisms, he said. And Deborah also, like, I think we all feel like that. But Deborah also had a question, and she says that thank you for your nice presentation. In the slide with the global distribution of black fungi, there were some countries on the map which seemed to not have black fungi. She might have missed this, but is that because there is no black fungi in those countries, or no sampling has been done in that region?
Mm-hmm. No sampling. I mean, we have covered the whole continents, but there are countries where we don't have sample, and some of fundings can be improved by future sampling, future survey, to sample more countries and more samples from the under-sampled locations. For instance, from South Africa, we have like dozens of samples, but of course, from that region, we need to sample much more, from China too. So we will look into that in the future global sampling. This was a pilot study.
Thank you. And Dave Berger from FABI also has a question. He said that does melanin in black fungi cause problems with DNA extraction? How do you overcome with this?
Yeah, as I said, the work in the lab with black fungi, it's beautiful but challenging at the same time. And as you rightly said, the melanin, the big amount of melanin, because these fungi are highly melanized, make difficult to disrupt the cell wall. So, for instance, for the DNA extraction is quite simple, because we were able to extract a good DNA quality, have a high concentration of DNA for the genome sequencing. But now we are experiencing some difficulty to obtain good and high concentrated RNA for the transcriptome, because the RNA is much more vulnerable than DNA, and the melanin is causing, it's great for black fungi, but not so much for us.
Yeah, exactly. I will just ask Carlton question because it is linked with Deborah's, but then I come to Thomas for this question. So Carlton from USA asking that what environments were black fungi collected to generate the distribution map? Were they collected from primarily extreme environments? He has also isolated some black fungi such as Chaetoreales, but always from plant tissues as apparent endophytes.
Yes, of course. In a future project, we will collect samples not only from soil, but even from plants, animals, and other natural environments and other polluted environments.
Hi, Claudia. Thank you for that. Yeah, it's always nice to see in what extreme environments fungi can actually survive and grow. I'm thinking about soil samples. It's obviously quite a big task to collect all of these soils from different places, and can get quite expensive to do as well. Do you have any advice how we can store the soil to best, I guess, number one, contain the DNA intact, and then secondly, so that we can isolate fungi in future from these soils? Obviously, it will be great if we can start sharing soils between projects that maybe don't overlap in time. So I'm just wondering, preservation of soil, if you have any tips.
Mm-hmm. Okay, so the first question, the answer is that we take advantage for global sampling from global network of collaborators, because as you said, it can be very expensive and very time consuming to sample the whole globe. So we have several global networks, and each collaborator provide soil for us. We have established a standardized protocol that are shared between collaborators, and even sampling kit. And in this way, we have samples collected in the same way and extracted in the same with the same procedure. But the conservation of soil can be at -20. It's okay for the DNA extraction, and even if you want to do some metabolomic or transcriptomic analysis, -20 is perfect for each analysis you want to do. And for fungi, from this global sampling, we didn't isolate black fungi. We did just some uncultivated molecular analysis. But if you want to isolate black fungi, it can be less easy than doing in Antarctic rocks, because the Antarctic rocks encompass well-organized microbial communities, but the biodiversity is very low. There are no dozens of microorganisms. So when we isolate fungi from Antarctic rocks, we have played with some isolate, like fungi, lichens, or algae. From soil, I think that we have to filter it what we want to look. If we want to look to cosmopolitan and fast-growing fungi, we need to do a grow medium. If we want to focus to the slow-growing fungi, we will need to filter the soil, washing with a specific procedure, and use some media that are specific for the slow-growing fungi. So according to what you want to focus on, there are several molecular procedures for isolation.
Yeah, thanks for that. The other question I guess I have, which we can maybe talk about later, is how do you get around all of the collection permits and Nagoya protocols associated with collecting all of these?
Yes, this is also challenging. I'm now writing the application for sampling permission for the Marie Curie fellowship to sample in the North America drylands. And you need to be very specific of what materials you are going to collect, the environmental impact, and everything. For the global sampling, when we are taking advantage for the collaborators, fortunately, those collaborators have other projects where we are collecting I don't know, for instance, plant or animals, and in the context of their projects, they can provide us with, for instance, soil samples.
Perfect, thank you.
Thanks, Claudia. I will hand it to Bernard to ask his question.
Claudia, thank you for a wonderful seminar.
Thank you.
So that Antarctic, what is it called, Death Valley, or valley where there was thought to be no life, and then these fungal communities inside the rocks are found. The divergence that you said, it diverged from other related life forms, hundreds of millions of years to more than 1 billion years ago. What does that correlate with that in those dates? And is that a unique situation? But I guess that area is quite isolated from the rest of the world, but other extreme environments, the deserts and so forth that you work in, or the hot deserts that you work in, would have aerial fungi blowing in there on, I assume, a more regular basis than in those Antarctic valleys. So do you, in these other extreme environments, or do you also see life forms that are that divergent in these extreme environments as you see in Antarctica?
Thank you for your question. This is the next step of our research. I mean, we did some preliminary evolutionary analysis to date the bacterial genomes we retrieved from Antarctic metagenomes, and we found that some bacteria species are from 3 million years ago, rightly when the Antarctica reached the South Pole. So the hypothesis is that some species, some taxa, evolved when the Antarctica reached the current conditions. But for other taxa, we found that evolved far away before the Antarctica reached the current conditions. So probably those taxa evolved somewhere in the world, inside a rock, and with evolution came to Antarctica. But for now, we did this evolutionary study with a selection of rocks and just from Antarctica. The next step is collect rocks from across the globe and did the same analysis to see if some lineages are shared between all extreme environments on Earth, or if some lineages are specific for a condition or another.
Interesting. Thank you.
Thank you.
I think if there is no other question, I will wrap up the seminar. Thank you again, Claudia, for the amazing presentation. We really
Thank you again
so
for
much.
inviting me. Thank you.
And good luck with your journey in USA, but we know that we will always be in touch, and I think Fabians even will see you more often than they think due to the projects that we will work together. And I am now handing, and thanks to Marcus and Mornay to making everything run smoothly. They are the behind the screen people mostly. And I now hand over to Bernard to close up.
Yeah, I don't need to say much more. It was really a fabulous journey to go with you, Claudia. I especially liked when you said it is currently not easy to travel to other planets. So, you know, assuming that in a couple of years it might be easier to study this in other places too. I really enjoyed it, and wish you all the best for your continued great work. Look forward to seeing you here and the results that will also come from the joint projects.
Thank you again for inviting me. Thank you.